Okay, let's unpack this. Forget towering redwoods or, soaring skyscrapers for a second. Imagine instead towers built by worms.
Right. Tiny microscopic worms.
Thousands of them piling together, creating these, well, living structures that almost defy belief.
And yet, it's precisely what's happening. Just on a scale we don't typically see. We're talking about nematodes. Yeah,
Nematodes.
incredibly common, often unseen roundworms. And they're engaging in this astonishingly complex collective behavior, basically right under our noses.
It sounds so bizarre, doesn't it? Like something out of science fiction.
It really does.
But the source material you've shared, this is a groundbreaking new scientific report. It gives us the very first direct look at this incredible behavior actually happening, not just in a lab dish, but out there in nature.
Exactly. They've actually found these living worm towers in the wild.
Which is huge, because seeing it in a lab is one thing, but
Right. What's really fascinating here is that while, yeah, self-assembly in nematodes had been seen in controlled lab conditions before, confirming that they actually do this in natural settings. And understanding the function of these towers, that was the crucial missing piece. This report really fills that gap.
So our mission today, then, is to take a deep dive into this report. We're going to explore this surprising discovery together, pulling out the most important, most intriguing pieces from the source material you've provided. We'll try to figure out what these nematode towers are, where they were found, how they seem to form, and maybe most importantly, what they actually do for the worms.
And perhaps even more broadly, what studying this unexpected collective behavior in these seemingly simple organisms can tell us about cooperation and dispersal strategies across the natural world.
Okay, all right, let's jump in. Describe this phenomenon for us. What exactly is a nematode tower?
Okay, so at its core, it's individual nematodes physically linking their bodies together. They stack or weave themselves into a vertical structure.
Like linking arms, but
Way.
with their whole bodies.
Yeah, sort of, yeah. Think of it as a living skyscraper, built molecule by molecule, except the molecules are worms.
And the big news from this report, as you said, is finding these actually happening in nature.
Exactly. This report provides the first direct evidence of these towers occurring in the wild. Outside of, artificial or semi-natural lab setups.
Just out there.
Yeah. Seeing them actively forming and functioning out in the environment, that's a significant breakthrough.
And they weren't found just anywhere, were they? Where did the researchers discover these wild towers?
They were found on rotting fruits, apples and pears, specifically in Germany, and also on a mushroom farm in the UK.
Ah, okay. So these are the kinds of nutrient-rich but temporary microhabitats that nematodes often colonize.
Precisely. Places they'd eventually need to leave when conditions change, like the food runs out.
Right. Were these towers made of just any nematodes that happened to be there? Or specific ones?
That's a key detail from the report.
Huh.
The towering individuals found in the wild were overwhelmingly from specific Caenorhabditis species. Yeah. Primarily an undescribed species the report calls CSE8 and also C. remini. They did also observe the famous lab species, C. elegans, forming towers under semi-natural conditions on that mushroom farm.
Interesting that it's primarily specific species in the wild. And what stage of life were the worms actually building these wild towers? Was it adults, larvae?
Exclusively the dower larva stage.
Dauers. Right.
Dauers are a specialized stress-resistant dispersal stage for these nematodes. They're basically built for surviving harsh conditions and traveling to new, hopefully better environments.
Okay, that makes perfect sense for dispersal then. I know nematodes also have another way to disperse individually, right? By sort of standing on an edge and waving in the air.
Yes, that behavior is called nictation. A single worm stands up on an edge and waves its body, probably trying to attach to a passing insect or some other animal for transport.
So towering seems like a group version of that.
Exactly. It seems like a collective strategy, sort of analogous to individual nictation, but involving the whole group.
So why build a tower as a group instead of individuals just doing their own nictation thing? What's the advantage?
Well, the primary proposed function, and it's strongly supported by the report, is collective dispersal. These towers are structures that help the group move to a new location, maybe more effectively than individuals could alone.
How do they achieve that? Do they, walk the tower across the ground?
Not quite. There are two main ways they seem to facilitate dispersal, according to the findings. One is simply by bridging gaps.
Bridging gaps?
Yeah, the towers can grow tall enough to literally span a gap and reach a new surface nearby. The report mentions a really striking observation in the lab where a tower managed to bridge a gap of about 3 millimeters
3 millimeters?
to reach the lid of a petri dish above it.
For a microscopic worm, that must be like traversing a canyon.
Absolutely. It shows the power of the collective, right? The second, and perhaps even more significant function, is foresy, or, hitchhiking. The towers are structured in a way that allows them to attach en masse to passing vectors, like insects, and be transported to entirely new locations.
Hitchhiking worm towers. That's just wild to picture. Did they find actual evidence of this happening?
They did. In the field, on those rotting fruits, they frequently observed towers co-occurring with adult and larval dipterans, that's flies, and also coleopterans, which are beetles.
Okay, so they were hanging out together, but co-occurrence isn't proof.
Right. It isn't definitive proof of interaction, but it's highly suggestive.
Okay, but seeing them actually get on a hitchhiker, that would be the real confirmation. Did they manage that?
Yes, they did this in the lab. They used an adult Drosophila melanogaster, the common fruit fly, and they observed that towers were indeed capable of detaching from the substrate and attaching themselves as a group to the flies as they walked by.
Wow, they actually saw it happen. The whole tower, or part of it, just latching on.
A group of worms from the tower, yeah. And they even simulated a passing animal with something artificial, didn't they? Just to see the tower's reaction.
That's correct, and it provided really compelling evidence. They used an inert glass pick to simulate a passing vector and gently touched the top of a lab-grown tower.
And what happened?
The towers responded remarkably. They actively grew towards the stimulus, attached the pick, and successfully transferred a whole group of individuals, about 110 worms in that specific experiment.
Whoa.
So this strongly supports the foresy function. And it shows that simple touch alone seems to be enough to trigger this attachment and transfer response.
That's incredibly cool. So confirming the natural occurrence, identifying the dispersal function, gap bridging, and hitchhiking, that's huge. But to really understand the mechanisms, like how they build these things, you need a more controlled environment.
Exactly. And that's where their lab work comes in. A major contribution of this report is the development of a reliable lab assay using C. elegans.
C. elegans, the lab workhorse. Precisely. They specifically chose C. elegans because it's arguably one of the most well-understood organisms on the planet. There's an enormous toolbox of genetic and experimental techniques available. This allows for really detailed study of the underlying biology.
Okay, so what specific conditions did they figure out were necessary to reliably get these worms to tower in the lab? What's the recipe?
3 key conditions consistently induce towering. First, a high density of individuals. You need thousands of worms crowded together.
Lots of worms. Makes sense.
Second, the absence of nutrients. Starvation, basically. If food is present, they don't tower. They're quite happy just eating.
Right. And third?
And third, the presence of some kind of climbing structure. Interestingly, they found a simple toothbrush bristle worked very effectively as a reproducible pillar for the worms to climb.
A toothbrush bristle? That just adds a touch of relatable weirdness to this whole thing. And this lab assay was really efficient, you said.
Extremely efficient. It reliably induces persistent towering within just 2 hours.
2 hours.
Yeah, which is a massive improvement over previous methods that could take days. So it makes it a powerful tool for controlled, long-term observation and, detailed experiments.
They also observed towers forming without a pillar sometimes, right? Just worms climbing on worms.
They did. Worms can build unsupported towers by climbing solely on each other, but the report notes these are much rarer, smaller, and definitely less stable than the pillared towers. The pillar seems to provide essential structural support for those really large sustained aggregations.
Got it. Now here's where the lab findings get really surprising, at least to me. The report reveals something unexpected about who is capable of towering.
Yes, this is a fascinating discovery, because while only dauers were found towering in the wild,
Right, the dispersal stage.
the lab assay showed that worms from all life stages, including day 1 adults, dauers, and even mixed stage populations, are perfectly capable of towering under these induced conditions.
Wait, all life stages, even the adults and non-dauers. But I thought nictation, that individual waving, was exclusively a dauer thing, linked to specific dauer development pathways.
That's precisely why it's so surprising.
Okay.
Nictation is tightly linked to the dauer stage and its unique neurobiology. The fact that non-dauer worms, which don't nictate, can readily tower, suggests that towering might be a much more generalized potential dispersal behavior.
Maybe something ancestral.
Possibly. or just a capability retained across different life stages, distinct from the specific dauer-exclusive nictation mechanism. It definitely raises the question of, well, why do we only see dauers doing it in the wild then? Maybe conditions are different or the trigger is more specific.
Interesting. Did they explore what factors might affect how tall these lab towers get?
They did. And there were some intriguing results there, too. They found that day one adults actually built significantly taller towers than mixed age groups or even the dauers themselves.
Hold on. The life stage optimized for dispersal in the wild. The dauers built the shortest towers in the lab. That seems counterintuitive.
It is, isn't it?
Yeah.
And it suggests potential differences in how different life stages might contribute to or benefit from towering. Perhaps dauers are optimized for quick collective attachment rather than maximum height.
Maybe height isn't the only goal for dauers?
Or perhaps the conditions for towering vary subtly in nature versus the lab. They also found slight differences between strains. The wild isolate CB4856 built slightly taller towers than the standard lab strain N2.
Which tells you?
Well, comparing these strains helps researchers understand if maybe laboratory adaptation, living in comfy lab conditions for generations, might have influenced the behavior in the standard N2 strain.
Fascinating details. Okay, so let's think about the structure and dynamics inside these towers. With potentially thousands of worms involved, how are they organized? Is it just a big jumble?
No, they observed a clear structural organization. As worms climbed upwards on the pillar, their bodies and heads were largely aligned. They showed what the researchers describe as polar order.
Polar order, so a directional arrangement.
Exactly. It's not just a random clump. There's a definite structure to it.
And these large towers, the ones with thousands of worms, can become quite substantial and dynamic.
Yes, on the pillars, they could grow well over a centimeter tall.
A centimeter? That's huge for them.
And display complex dynamics.
Yeah.
The report describes large towers forming these unsupported arms that would, explore the surrounding 3D space.
Exploring arms made of worms.
Yeah. These arms might sometimes collapse, but the worms would often reorient and just reintegrate back into the main tower, sometimes branching out again.
And I remember you mentioning one of those exploring arms was what actually bridged that 3-millimeter gap earlier.
Precisely. An arm extended from the main tower, reached across the gap to the petri dish lid above, and formed a stable structure that allowed worms to travel from the base all the way up and over.
That's just a powerful visual of their collective building capability. Incredible.
It really is.
Okay, so this is such a complex, coordinated structure involving thousands of individuals. It makes you instinctively wonder if there's role specialization. Are the worms at the bottom doing different jobs than the ones at the top? Are some stronger or maybe more reproductively fit in saving themselves?
That was a key question they investigated.
Yeah.
Is there role specialization within the tower? You'd almost expect it, right?
Easily.
But the surprising finding from the report is they found no clear evidence of specialization.
Really? None at all. That kind of challenges the idea that complex structures require different roles, doesn't it? How did they test for that?
They performed several specific experiments using worms sampled from different positions within or around the tower from the very top, from the base,
Yeah.
and also from non-towering worms just hanging out outside the structure.
Okay. And what did they measure?
They tested things like reproductive capacity by measuring brood size, basically, how many offspring they produce later. They looked at muscular strength by measuring their thrashing rate, which is how fast they wiggle their bodies in liquid, and various locomotion attributes like speed and how often they turned.
Right. And the results? Were the base worms stronger? Were the top worms maybe sacrificing reproduction?
Across all those tests, they found no significant differences between worms sampled from the top, the bottom, or outside the tower. They showed similar reproductive rates, similar strength based on thrashing, and similar general crawling patterns.
Huh.
And furthermore, when a mixed stage tower dispersed onto that glass pick we talked about, the population that transferred was also mixed. There wasn't any sorting based on life stage during dispersal either.
So what does this tell us then? For these largely clonal populations they used in the lab, it seems like every individual is capable of performing pretty similar functions regardless of their position in the tower.
Exactly. You might expect a complex structure like this to require different roles for, say, stability or efficiency, but in this system, it appears not to be the case, at least for the characteristics they measured.
It suggests maybe a simpler cooperative mechanism than what you might see in, I don't know, a complex insect colony with distinct castes.
That seems right. Though the report does wisely note that future studies, perhaps using genetically diverse populations, could potentially reveal more nuanced competitive or cooperative dynamics based on individual genetic makeup. Maybe there's stuff happening they didn't measure.
Always more questions. Okay, stepping back then, what's the broader significance of this report? It seems like it does a lot more than just describe a weird worm behavior.
Oh, absolutely. It fills a crucial gap by definitively confirming that this fascinating collective behavior actually occurs in nature and serves a vital ecological function dispersal, especially via hitchhiking to find new patches of food.
Which was suspected but not really proven in the wild before.
Correct. And crucially, by establishing this reliable laboratory model system using C. elegans, it provides a powerful, tractable tool for future research.
And what kind of doors does that open up for future research? Where does this lead?
Well, this system is just ideal for diving deep into the mechanisms behind this behavior, the genetics involved, the neurobiology, how they sense each other, and the environment to coordinate building the tower.
The how.
The how, exactly. It's also great for studying the ecology and evolution of collective dispersal and cooperation in general. These are areas that are often incredibly challenging to study in larger or less manipulable organisms.
Easier to control thousands of worms than thousands of birds, I guess.
Something like that, yeah.
And they can compare this across different types of nematodes too, right? Not just C. elegans.
Exactly. The methods they've developed can potentially be applied to other nematode species, and this would allow for fascinating comparative studies.
Like seeing who towers and who doesn't.
Right. Understanding what factors, maybe environmental cues, genetic predispositions, might have driven the evolution and diversification of this collective powering strategy across the incredibly diverse nematode phylum.
Yeah.
There are thousands of nematode species out there.
It really is remarkable when you stop and think about it. Thousands of these tiny worms that, we usually only picture wiggling individually in soil or something, are actually cooperating, aligning their bodies, building living structures much taller than themselves, and then traveling together to new worlds.
It fundamentally challenges our assumptions about simple organisms, doesn't it? It highlights that complex collective behavior and sophisticated strategies aren't limited to the insects or vertebrates we often focus on.
Yeah.
They can exist in these microscopic worlds and serve absolutely vital functions like finding the next meal or surviving environmental change.
So what does this all mean for you, the listener, this deep dive into nematode towers? It really makes you wonder, doesn't it? What other unseen collective behaviors, driven by surprising cooperation among tiny, overlooked creatures, might be happening all around us in the natural world? How widespread could this towering strategy, or maybe similar forms of collective dispersal, actually be across the vast unexplored diversity of nematodes and perhaps other seemingly simple organisms just waiting there to be discovered?